last lecture for this first half of the class. It's a topic that I think is really important to discuss, in part because it kind of cuts through a lot of the issues we are dealing with over the course of this entire first half of the term, and also just because I don't think it would be appropriate to teach a class on computational molecular biology and not say something about this topic of phylogenetics. So this doesn't quite fit neatly into any of the individual topics we've discussed over the course of the different weeks of this class, so I just wanted to kind of wrap up with a lecture on this. This is quite a big area. There's a lot of computational theory and mathematical modeling behind phylogenetics, so I wanted to give you kind of a whirlwind for a lot of the different options that are available and not spend too much time on any one particular issue of this. And I'll give you some pointers later where any of you who really need to do serious phylogenetics work can learn more about it. But basically, when we're talking about phylogenetics, what we're talking about is evolutionary trees. So phylogenetics is the study of ancestral relationships. And usually what we're assuming when we do a study in phylogenetics is that we are trying to build an evolutionary tree. We have some set of data, typically data about distinct species, although not always. And what we're interested in doing is somehow computationally inferring an evolutionary tree among these different elements of our data set, or organisms, or as we'll see, sometimes other levels of the diversity that we're interested in. And I think there are a lot of important reasons why this is a topic you should really know something about if you are going to be doing any work that involves computational biology. In part, I think phylogenetics is an important area just because we are often curious about evolution. If you're doing basic research in biology, everything is related to evolution. Having good models of how evolutionary relationships come about, how organisms are related to one another, is fundamental to a lot of questions in the development of biological systems. Beyond that, evolution can help us understand function. So there are a lot of techniques by which we can use an evolutionary tree or a model of evolution to help us understand what pieces of a genome are functional, which ones are being selected for, or in some cases selected against by evolutionary pressures, how different functions might be appearing over the course of evolution. So we can learn a lot by just seeing how different organisms are related to one another. Beyond that, we've kind of gotten a flavor in a few different applications of how evolutionary trees can help us with many other kinds of computational inferences. So this falls more or less into the general area of comparative genomics. And you may remember a version of this figure that we've seen previously when we talked about some of the ways we use whole genome information. So this is a figure showing sequence conservation for a small region of the human genome relative to various other organisms. And by seeing that there are spikes of conservation, so regions that are very highly conserved, we can often get hints about functional relevance of those regions of the genome. So a lot of the kinds of techniques we've seen over the course of this half term have versions that have been made more powerful and more effective by bringing in these kinds of comparative genomics ideas. So we saw briefly an example of that in the context of gene structure determination, where being able to look at multiple related organisms allows us to more accurately predict gene structures in each individual organism because of the patterns of conservation of these sorts of structures across organisms. And a lot of current work in computational biology is doing more sophisticated versions of this. So by using detailed evolutionary models of many organisms, we get stronger power to do all sorts of inferences that we might want to do about relationships between sequence, function, and so forth in individual organisms. Finally, there are some specialized uses of these in various contexts. So we saw an example of a special kind of phylogeny, a pedigree, for use in linkage analysis, one of the techniques that we've seen for associating genotype with phenotype. And phylogenetics also becomes quite important in association studies. So the other major technique we saw for associating genotype with phenotype, in particular, understanding how different organisms in a population are related to one another helps us control for some of the confounding factors, so some things that can kind of confuse our analysis if we're not aware of those relationships and trying to do those kinds of studies. So this is all kind of motivation for why I think it's worth knowing about evolutionary tree building. Are there any questions about any of that? All right, so if we want to do phylogenetics, we need a source of data, and there are a lot of different kinds of data you can apply to phylogenetic inferences. I mentioned a number of these over the course of the term. So phylogenetics actually predates computational biology. People were building evolutionary trees before we had computers to build our trees. And in the early days, this was commonly done through morphological data. So we would look at observable features of an organism, like does it have wings or feathers or scales or fur? Does it lay eggs? Is it warm-blooded? How many toes does it have? All of these kinds of things that presumably are relatively hard to evolve and don't appear very often in evolutionary histories can give us information suggesting which organisms are more or less closely related to one another. That's still used in some contexts today, although it's mostly been displaced by sequence data. So for the most part today, when we talk about phylogenetics, we're trying to do inferences from sequences. Sometimes this is going to be DNA sequences, sometimes amino acid sequences, but some kind of sequence, just because we have so much more data in the form of sequences, and it lets us make our inferences much more accurately. Can anyone think of why we might still be building evolutionary trees from these older kinds of data? What might be an application of that? Well, it still comes up. Oh, is there
I was going to say compare it to the sequence data to see, I guess, like the accuracy or whether the trees were converged upon separate sequences or from a common one.
Yeah, it can be useful to compare different kinds of data, so to have morphological and sequence data and see how much they're in agreement with one another. One place this still comes up quite a bit is people are interested in evolutionary trees of extinct organisms, and extinct organisms you often can't get your hands on sequence data. So people want to know how dinosaurs evolved, let's say. It's debatable. There's either little or no sequence information available on dinosaurs, so you have to kind of rely on these older sorts of things. There are lots of other kinds of information we can use, some of them quite important, some more specialized. So we saw genome rearrangement, so rearrangements of organization of genes on a genome, and this is quite important for understanding evolution at very long time scales, where so much time has elapsed that conservation on the sequence level is simply lost. But there are other examples of this. Protein domain structure is something you can use for evolutionary studies that's more specialized. You can use three-dimensional structure as a source of information about evolution. You can study evolution of networks. So like we saw last week, we can see how networks are evolving over time and build phylogenies based on those. So almost anything that changes slowly enough in organisms over many generations can be a marker for different kinds of evolutionary studies. But for the most part, I'm going to assume we're looking at sequence data because that's mostly what people are using to build evolutionary trees today. So there are a lot of variants on phylogenetics, and I don't have time to go through every instance or every special case of phylogenetics where people are doing research today, but I did want to talk about some of the high-level ideas behind this and show you a few examples of different techniques that are representative of these different classes. At the highest level, people generally divide phylogenetic methods into distance-based versus character-based methods. And I'll say more shortly about what that means. But basically, distance-based methods tend to be things that are very good for looking at instances when you have very large amounts of data, so whole genomes or large portions of genomes to look at. Character-based methods tend to be better when you need more detail on the evolutionary process and you're not trying to make sense of as much data at a time. Within each of these, there can be variations on how we measure evolutionary distance or how we pick characters. So characters being simply traits that vary over time that we can use to make inferences about evolution. There are different versions of trees we might be interested in. Most commonly, when we think of phylogenetics, we're thinking of what's called a species tree, where the nodes of our tree would represent species, and we're trying to understand how these diverge. But there are different levels of resolution, such as, for example, a gene tree, so studying evolution of particular families of genes within a set of evolving species. You can also do phylogenetics interspecies or intraspecies. So there are specialized methods for the case where you're looking at evolution of, let's say, human beings over the course of our history, where you have not clean separated species, but intermixing populations. There are different objective functions, which refers to kind of the mathematical model we use to decide how relatively good different trees are for a data set. So in some cases, we'll see a relatively simple version known as parsimony, or more or less equivalently minimum evolution. That's one way of posing the problem. But in other cases, we'll be looking at more sophisticated probabilistic variants, such as what are called maximum likelihood or Bayesian models. For any of these, there are also different algorithms that might be of interest to us. So a lot of phylogenetics is done with very specialized, fast heuristic algorithms that will not necessarily get you the best possible tree for your data, but will do it quickly. There are other classes of algorithms you might use when you really want the best tree and you're willing to put in a lot of time for that, as well as various statistical methods we can use to sample over sets of possible trees and maybe not look at one best, get an idea of what's in common among many pretty good trees for a data set. Now this is a large field in part because it's a difficult field. So it's difficult to do good phylogenetic inferences. Part of the reason it's difficult is because of basic computational issues, and in particular the fact that pretty much any reasonable version, any reasonably realistic version of phylogenetics is going to be computationally intractable. So over the course of this term, I've mentioned a few instances of intractable problems. Does anyone remember what an intractable problem is?
Like the traveling salesman problem, there's no algorithm that's finite that you get to an answer with, or a definite answer.
Yeah, so intractable would mean that there's no algorithm that runs reasonably fast that will guarantee you the best possible answer. And reasonably fast is usually a more technical definition, means it runs in time that is less than exponential in the amount of data you're trying to reconcile. So there is a formal technical definition, but generally intractable we would think of as being the problems where you're not going to be able to get the best possible answer reliably, except for very small data sets. And pretty much any version of phylogenetics falls into that category. We also have to deal with the fact that uncertainty is often high in our inferences. It's not so unusual if we might be trying to build an evolutionary tree, and there's some organisms where we have a lot of data cleanly separating them, but in other instances the data is very ambiguous, and we need to take account of that. And we have to take account also of the fact that our models tend to be highly simplified. So evolution is a complicated process, and usually when we're building evolutionary trees, we're making a lot of simplifying assumptions. So, for example, a tree will generally assume that we don't have horizontal transfer, so genetic material moving between different organisms or different species except through reproduction. That's not exactly true, and in some applications of phylogenetics, it becomes quite important to take account of the fact that these things occur. So again, we often have to pay a lot of attention to our models and the implications of the models for the algorithms we can use. So all of this is why, in a sense, there are so many methods for phylogenetics, because it's a hard problem, and we often really need to deal with the details that are hard to handle in this problem. So any questions at this point? All right, so let's start looking at some of the actual methods. And I'm going to start by looking at distance-based phylogenetics. So in distance-based phylogenetics, we would generally assume that there is some pre-processing step that is letting us estimate evolutionary distances. So roughly speaking, the amount of time or the amount of mutation that elapsed between two given organisms. And we would then build the trees using these evolutionary distances rather than our raw data. So, for example, our raw data might be a set of sequences, maybe a set of nucleotides in some conserved region, some gene sharing among our organisms, and then we would try to estimate how evolutionary distant these different sequences are. There are various ways you could do that. An alignment score can be one way of doing that. We might say that if we think mismatches occur at some evolutionary rate, and gaps appear at some rate that's maybe 5 times slower, then an alignment score of saying that a match costs 0, a mismatch costs 1, a gap costs 5, can give us a rough measure of evolutionary distance between sequences, and we can then look at all the ways of pairing up different sequences and get an estimate of how far apart they are in an evolutionary sense. That is commonly encoded in what's called a distance matrix, and a distance matrix is simply a way of saying between any pair of the taxa in our data set, so any pair of the species we're trying to reconcile, what is the distance between them? So if we want to know the distance between, let's say, taxon 2 and taxon 3, we would look at either row 2, column 3, or row 3, column 2, and we would see the distance between them is 3. So for distance-based phylogeny, we would assume this matrix is the thing we're trying to use to infer the evolutionary tree. And there are different ways of doing this, which will mostly come down to throwing in some additional assumptions about how evolution works, and trying to find a tree that is consistent, or at least as consistent as we can get, with the distance matrices. So does this make sense to everyone? All right, so the simplest algorithm for doing this is an algorithm called the unweighted pair group method with arithmetic mean, or UPGMA method. And the UPGMA method is a method that proceeds from an assumption that evolution occurs at equal rates on all lineages of a different set of species over any given point in time. This is known as the molecular clock hypothesis. It basically says that if you look at 2 organisms evolving over the same span of time, we would expect to get about the same number of mutations in each of those organisms over the same amount of time. That's often not really a true hypothesis. It's not really entirely accurate, and there are lots of reasons. You might get different rates of evolution along different lineages, but it's a model, and it's a model that lets you come up with algorithms. If you assume that, then the algorithm works as described in this pseudocode, so this kind of high-level description of what's going on. And basically the idea is that we take our taxa, we take our distance matrix, and we just repeatedly merge the 2 closest taxa. So we find the 2 that have the smallest distance, we join them together under a new parent node, which is assumed to be their common ancestor. We do some calculations to estimate the distance between that parent node and the 2 children we just joined, as well as between the parent and everything else in the tree. And we just keep doing this until we're down to 1 node, which then becomes the root of the tree, so the putative common ancestor of everything in the tree. So that's the pseudocode. And I think to understand how this works, it helps to look at an example. So let's say we have this distance matrix, and this is an illustration of our 4 taxa in this example. So we're assuming taxon 1 is a distance of 1 from taxon 2, 2 from taxon 3, 4 from taxon 5, and so forth. And if we want to apply the UPGMA algorithm, what we would do then is look at these pairwise distances and find the 2 taxa that are closest among all the pairs of taxa. So which 2 would those be? Well, the distance matrix is telling us how far apart each pair is, sitting here or here. So either row 1, column 2, or identically row 2, column 1. So that's telling us taxa 1 and 2 are a distance of 1 apart. So those are the 2 closest things. So we would merge those and create a parent node above them. Because of the molecular clock hypothesis, what we're going to do is take that distance of 1, and we're going to attribute half that distance to the branch from the parent to node 1, and half of it from the branch from the parent to node 2. So we know we should have a distance of 1 between nodes 1 and 2, and we're saying you get half of that going from 1 up to the parent, another half going from the parent down to node 2. So we've got that distance there, split half and half like the molecular clock hypothesis. And we then need to estimate what the distance is between now this merged subtree and the remaining nodes in our tree. And the way we do that is simply to say that we estimate the distance of any of these other nodes to this new parent to be the average of the distances to the children. So the distance from 3 to this parent, we will say, is the average of the distance from 3 to 1 and 3 to 2. The distance from 3 to 1 is 2, from 3 to 2 is 3. So we say the distance from 3 to this parent node is the average 2.5. We likewise would say the distance from 4 to the parent is the average of the distance from 4 to 1 and 4 to 2. Those are both 5. So the distance from 4 to 1, 2 is 5. And we now have a tree that has 3 nodes. So node 3, node 4, and the 1, 2 parent node. And then we repeat the process. So what should we join together next? We effectively have 3 nodes here, so we're looking for the smallest number in the submatrix, and it's the 2.5 here. So either the connection from 3 to 1, 2, or symmetrically 1, 2 to 3, that would be the thing we would join next. So we're going to place a parent node above 3 and this 1, 2. So that becomes this new parent node of 1, 2, and 3. And we're going to say the distance 2.5 that we had from 3 to this subtree gets split half on this branch, so 1.25, and half on this branch. And since we already had a distance of 0.5 here, we need another 0.75, so that the distance from 1 to 3 will be 1.25 going up this branch, another 1.25 going down this branch. So a total of 2.5, and the same for 2. So we're not exactly getting the distances we wanted between 1 and 3 and 2 and 3. This should have been 2, this should have been 3. Instead, they both get averaged 2.5. So we're not getting a tree that will exactly match our original distance matrix, but we're getting as close as we can subject to the molecular clock hypothesis. At this point, there's just one thing to do. We've got this subtree represented by this node, and we've got one node left. We have to join those together. And since we had a distance of 6 between those, we attribute half of it to this branch and half to this branch. So if you follow the edges down here, they will add up to 3, whatever path you take. So that basically is the UPGMA algorithm. And that's probably the simplest phylogenetic tree algorithm you can come up with. So any questions about that? So we had actually seen this algorithm for a slightly different application before. Can anyone think of where we saw this same algorithm before?
Is it like the clustering algorithm when we were merging data points?
Yeah, so this is the same as the hierarchical clustering algorithm we saw. So if you measure your distance between points in your clustering, if you treat that as an evolutionary distance, then you get exactly this algorithm out. So it's essentially treating evolution as a kind of clustering problem. And it gives us a simple but often not great algorithm for doing that. So any questions? So a more popular algorithm, one that has somewhat better theory behind it and often comes up with better trees, is something called the Neighborjoining algorithm. And the Neighborjoining algorithm says that instead of trying to use this molecular clock hypothesis, we will try to find trees that are selected according to a different criterion called the principle of minimum evolution. And the principle of minimum evolution simply says that among all the possible trees we could put together for our data set, the best tree is the one that involves as little mutation as possible. So if you assume mutations are relatively rare, then comparing any 2 trees, the one that involves less evolution is the better tree. And that's the assumption here. We don't have time to go into detail about all the math behind it. It's more involved than the math in the UPGMA algorithm. So I'll just say at a high level, we would often think of this as starting with what's called a star phylogeny, which basically means throw in a root and just assume every node is directly connected to the root, and then repeatedly subdivide this by doing some fancy arithmetic to kind of correct the distances so that we can look at the pairwise distance between any pair of nodes adjusted relative to the distance to all of the other nodes. And I know I'm not explaining why these formulas do that. Just take my word for it that they do this in an interesting way that lets us get the tree consistent with this principle of minimum evolution and consistent with our distance matrix. And just to show at a very high level how that works, we would start with a data set like we had previously. We'd start with a set of taxa. We'd put these into a star phylogeny, compute modified distances, and then repeatedly sort of break this apart until we get down to what's called a binary tree. So a tree where every node either has 2 children or 0 children in the tree. So we would look for the 2 closest nodes in our corrected distances. We would get maybe these are the 2 closest, and we would end up assigning a parent to those 2 nodes in a way that allows us to minimize the total cost of evolution between those nodes, as well as connecting them to the rest of the tree. So you can end up with something where we don't have the molecular clock hypothesis. You might end up inferring that the parent of nodes 1 and 3 is actually the same as node 1 and very different from node 3, because that can give you an easier way or a cheaper way of connecting to the rest of the tree. And we just keep doing that. We can get other nodes inferred here, other ones, and eventually we can rearrange this to look like our more familiar evolutionary trees and end up with an answer from the neighbor-joining algorithm. So in this algorithm, we will actually get the pairwise distances preserved exactly as they are in our original distance matrix. We want the distance between nodes 1 and 3 to be 2, and if we look at the distance along the edges here, we get a 0 plus a 2, so that's correct. From 1 to 2, we want a distance of 1. If you add these up, you get 0 plus 0.25 plus 0.25 plus 0.5, so that's 1, and 1 to 4 should be 5. 0 plus 0.25 plus 0.25 plus 4.5, that is 5. And if we look at any of the other pairs, it will add up the way it should add up. So this is going to give us an algorithm that will get something that matches our original distance matrix and does it in the way that minimizes the cost of evolution. So it's a fast algorithm, it has a reasonable motivation behind it, and it will give us something that matches our distance matrix. But it does have some significant drawbacks. And probably the main one is that it's possible for this to give biologically impossible trees. So, for example, it's possible for this to infer a tree where some of our edges have negative weight. Of course, you can't have a negative amount of evolution. Things don't go backwards in time, but that can come out of this kind of algorithm. So, usually if people are doing distance-based phylogenetics with reasonably hard data sets, this is still the thing you would use. And for most data sets, it does a reasonable job. But it's not a perfect algorithm. There are more sophisticated things people would often use if you have the compute time or if you know enough about the phylogenetics to do something more sophisticated. So any questions about that? All right, so there are a lot of other distance-based metrics, and many variants of the methods we've seen, lots of other heuristics like the neighbor-joining idea that people use. There are ways of taking these existing algorithms and making them more realistic. So you can solve for the principle of minimum evolution and throw in the constraints that you can't get negative weight edges or other things that are unreasonable, but that makes it a much harder computational problem, and you need more advanced algorithms to solve it. There are other whole classes of methods I'm not talking about just because we don't have time today. And a lot of what's actually done today involves probabilistic models. So rather than trying to find one best tree, we might set up a probability function, so a likelihood function of how likely different trees are for a given data set, and we can either optimize for that likelihood function or sample over it to try to see the space of possible trees consistent with our data set and get some more useful inferences about what those trees have in common, what they don't have in common, and what our uncertainty is of the tree building process. But that's kind of a quick tour of just a couple of methods that we would use for distance-based phylogenies. And again, distance-based tends to be a good thing if we have a lot of data to support our measurements of evolutionary distance. They'll tend to be very inaccurate if you only have a few variants to look at or you're looking at very short amounts of time. But if you've got whole genomes and you're looking at millions of years of separation, they tend to be more tractable methods for doing this. The alternative is character-based phylogenetics. And in character-based phylogenetics, we would try to work directly with our genetic variants. So maybe we're looking at a conserved region of genome. We find that there are some genetic variants in the unconserved sites in that region we can use to try to figure out how different organisms evolved from one another or from common ancestors. And in character-based phylogeny, you try to directly use this data and find a tree that explains how the sequences evolved over different edges of the tree. So we would assume in this kind of approach that each node in our tree is annotated with a version of these sequences. The leaves typically would be the sequences we actually observe, and then we would try to infer the sequences of ancestral nodes in the trees, the common ancestors, and infer which mutations are occurring along which edges in the tree. So you get a lot more out of a character-based analysis. It's more informative about what the ancestors looked like, what the actual process of evolution was, but the trade-off is that in doing this, you're creating a much harder computational problem. And the more of these sites you're looking at, the harder it gets. Depending on the method you use, it's often exponentially harder in the number of sites, which is why if you have enough to get a good evolutionary distance measure, you often wouldn't use these kinds of approaches. So that is basically what a character-based method is at a high level, and I also want to run through some examples of that. Let me pause again for a second and ask, are there any questions on what I mean by a character-based phylogeny? All right, so these characters could be morphological traits. It could be your character is the appearance of fur or feathers or egg laying or whatever, but normally these are going to be genetic data like this. And in a lot of these applications, the simplest thing to do is to use what's called maximum parsimony, which is a kind of discrete version of the principle of minimum evolution. So maximum parsimony says that among all the trees we can consider, the best tree is the one that involves the fewest mutations. Basically, you want to take the sequences you have and figure out a way of linking them up so that there is as little mutation along the edges of the tree as possible. That can be a very good assumption if we're looking at slowly mutating variants or short amounts of time. It's not such a good assumption if we're looking at very long amounts of time or very rapid mutation processes, but people will often use this because it tends to be the simplest of these character-based methods and the one that's most amenable to fast algorithms. So we can come up with an algorithm for a relatively small tree like this. It would search all the possibilities, and then we could say that maybe a tree with 5 mutations is the optimal here. Maybe we can find one that links together these sequences with just 4 mutations and would be somewhat better. Or maybe we find trees with 6 mutations that are worse. That's the general idea behind this, though. From a computational perspective, it's relatively easy to generalize to what's called weighted parsimony, which is basically the same thing, except that we charge different amounts for different mutations. So in a weighted parsimony, we might say that maybe A to G or C to T mutations are less costly than A to C or A to T or G to C or G to T. So that's one way of doing weighted parsimony. You can also say that maybe different sites have different weights. So some of your genetic sites may have a very high propensity to mutate. Some may be under much stronger conservation and mutate slowly, and you can charge different amounts for mutation in different sites. Computationally, it's generally not too much harder to do these weighted versions, but it's still a fairly simplified model. Even for these models, when you get past more than a few taxa, you really can't afford to look at every tree. So what is often done is a variety of heuristic methods that can do a pretty good job on most data sets, even for fairly large trees. And typically these work with a kind of strategy called a Monte Carlo method. And a Monte Carlo method basically just means try lots of random things and see if they give you better trees. So a common example of a Monte Carlo move in tree building is what's called a prune and regraft move. We would take our tree and we would pick some random subtree, pull it off and glue it on somewhere else on the tree, and ask, did this give us a better tree, so a lower cost tree? Usually it won't, but sometimes it will. And we can say that if it does, we will preferentially keep the new tree. If it doesn't, we'll preferentially go back to the old tree and try something else. So basically, most of what people do for this phylogeny building involves combinations of these sorts of move sets, and there's a lot of kind of tricks of the trade. There's some theory behind this, but a lot of it is just figuring out what works through a lot of empirical trials. And that's most of what is actually done for maximum parsimony tree building, as well as some of the more involved methods we'll get to shortly in real practice, at least if you're looking at more than a fairly small tree. Any questions at this point? In some cases, you can get to reasonably large trees and actually try everything. So with these maximum parsimony approaches, there are ways to figure out, in some cases, if the tree is not too complicated, what the absolute best tree is. Branch and bound methods are one example of that, a kind of search strategy to efficiently search through the space of all possible trees. There are more sophisticated strategies that use more advanced computer science, but sometimes these things are appropriate, but usually you're not going to get to big trees with that sort of method. Often people will move to the probabilistic sorts of methods, maximum likelihood or Bayesian modeling, in which instead of using parsimony, we will set up a probability function, so likelihood function. We've seen likelihood functions in a few contexts before here. In this case, we would say that our data would be our set of characters, or strings of characters characterizing our taxa, and our model would be the evolutionary tree. And then you could compute a probability, so a likelihood function of a particular tree producing the data you get to observe. In a simple version, it might be as simple as multiplying over contributions of different branches in the tree. So what is the probability of having this particular branch and having it output of one mutation of an A to a T? You can get a lot more involved with this, talking about prior probabilities of different topologies of the tree, different branch lengths, different rates of mutation. So these approaches can get quite involved in terms of the model of evolution, and that's a lot of why people are interested in them. They can also give you a lot better handle on uncertainty in your trees, so figuring out if you are looking at a tree that is really strongly supported by your data, one that's more weakly supported, or something in between. I will point out you can get that kind of information even from the simpler maximum parsimony type approaches. And usually with phylogenetics, you don't just generate one tree, you do some kind of study that will let you get a handle on uncertainty in your analyses. If you were doing a maximum parsimony tree, you would commonly do what's called bootstrapping, which basically means try lots of random subsets of your data, see which trees are produced by these different subsets, and try to figure out what those have in common to see what aspects of your tree are strongly supported by the data, which are very uncertain. If you're doing these Bayesian type approaches, then often that comes directly out of the methods. You sample over your likelihood function, and you can figure out which trees are well supported and which features of trees are supported by a large fraction of the likelihood probability. But this is something I mentioned in this context. It's actually something done quite a bit in many other kinds of computational biology analyses. So lots of the things we've considered over the course of this term, really in more advanced practice, you would want to do this kind of confidence estimation or uncertainty estimation in addition to just finding your one best answer. Any questions about any of this at this point? People generally think of those two variants of building species trees in either distance-based or character-based. We have various methods for solving those. But it is worth mentioning that there are a lot of versions of phylogenetics that come up in more specialized uses, each of which has its own body of theory and methods that have been developed to serve it well. So an example of this is consideration of evolution at the gene level. So we often look at species trees, but in some cases we have to look at evolution of families of genes. If you look at something like a human being, many of our genes will come in families where we'll have multiple homologous versions of the same gene. And if we want to understand how a human being is related to a mouse, let's say, we need to understand evolution at that level. We need to understand how a common ancestral gene might have been duplicated on the transition from human to mouse, or versions may have been gained, versions may have been lost. And this is kind of illustrating that by showing how the species tree, represented by the thick gray edges, may have kind of embedded within it a more complex process of evolution of particular families of genes to get to how the homologs of those genes are distributed in different modern organisms. And for a method like this, or a problem like this, where you have to worry about these extra operations like horizontal transfer, gene duplication, gene loss, you basically need to come up with a whole different body of theory to do your phylogenetics on these kinds of applications. Interspecies phylogenetics is another example of this. So a lot of what we're interested in understanding genetic variation data has to do with problems of evolution within single species. And that creates a lot of complications as well for your phylogenetic methods. So if our sequences represent, let's say, a set of genetic variants in the people in this room, then it may be that some of us have a pattern of genetic variants that is actually identical to the common ancestor of others of us. And that would be something we have to be able to infer in our trees. You may have other complications, like the fact that when you're looking within a species, you have different populations intermixing with one another, and you get processes such as recombination of genetic material that we would generally ignore when you're looking at a species tree. That creates other kinds of complications, and you need other kinds of algorithms to deal with it. For example, one of the algorithms we would often use for intraspecies phylogenetics is something called a median joining network algorithm. And a median joining algorithm is another sort of heuristic approach that tends to be a good thing to do if we have trees where our common ancestors may still be present in our data set. So this is some pseudocode, but at a high level I can kind of illustrate graphically. The way this would work would be that we would take our nodes and we would put together what's called a minimum spanning tree. A minimum spanning tree basically means link together the actual nodes we have, and only those nodes, without any inferred common ancestors, to minimize the evolutionary distance between the different nodes. And that's actually an easy computational problem. That's something we can do very quickly. But what we then try to do is figure out if we're missing nodes in this tree. And we do that by picking triplets of nodes, taking the average of those, what's called the median, and seeing if we can lower the cost of our tree by sticking the median into that tree. Here we have a tree that requires 5 mutations to get among these organisms, and we can try taking, let's say, the median of nodes 1, 2, and 3. That would have this sequence TAGC, and we stick that into the tree in the best possible place. If we do that, we can see we still have 5 mutations, so we haven't actually improved our tree any. So we would say that probably isn't the median that was an ancestor in the tree. In some cases, a median, like median of 1, 3, and 4, would actually give us a higher cost tree. It ends up hanging off of what was a branch in the tree, so we've made our tree worse by introducing that median. But sometimes we find a median that can actually lower the cost of the tree. So assuming that 2, 3, and 4 have a common ancestor actually lets us get a tree that requires fewer mutations to link everything together. So we would say we'll throw that into our tree and then proceed with our further analysis. This isn't guaranteed to give you the best possible tree, but it will do a reasonably good job and is an approach people would often use for dealing with these intraspecies problems. More complicated kinds of things are needed if we want to deal with the process of recombination. So if you remember, we've talked about recombination in the context of linkage studies. So recombination is the process by which genetic material we inherit from our parents gets mixed together into these kinds of patchwork chromosomes we would pass on to our children. And that creates a complication in that it means that a particular genetic sequence that we inherit from our parents might actually have multiple ancestors in our grandparents' generation. When you start taking account of those things, evolutionary tree building actually becomes a misnomer. What you're inferring is not really a tree, it's what's called a network, because you may have nodes that have multiple ancestors within the network. And that requires whole other classes of algorithms that become much harder to deal with and need more sophisticated theory, but are able to solve variants of these problems, at least reasonably simple variants of them. So these are active areas of research. People are still trying to develop better methods that can deal with larger numbers of taxa, larger data sets. But this, I hope, gives you some flavor of at least some of the complexity that goes into these specialized kinds of problems. And there are lots of other versions of this, numerous other specialized kinds of phylogenetics that different people work on. My own lab, for example, works on cancer evolution, and there, because tumors evolve in different ways than organisms, you need a whole other class of theory to build evolutionary trees for the kinds of mutations you see in cancers. There are also lots of other more sophisticated algorithmic methods that have been applied to these kinds of problems. So various other kinds of search heuristics. There's a huge body of theory of good search heuristics that are available that people have applied. These Monte Carlo kinds of methods, as well as alternatives, or called approximation methods, more advanced algorithmic techniques like a strategy called linear programming, and lots of other things you would learn about if you go into more advanced computer science that we can cover here. Likewise, there's a huge body of theory on the probabilistic methods, so ways of posing these sorts of Bayesian and maximum likelihood models, ways of sampling over spaces of solutions. That in itself has its own huge body of theory that you could spend many years studying if you want to learn the best ways of doing that sort of thing. And if you really want to know more about this, what I would say is take a whole class on this. Here, at least here at Carnegie Mellon, you can take a class specifically on phylogenetics, so you can get not sure now if at this point it's a half term or a full term, but you can take an entire class on this topic, and even that is not going to cover anywhere near everything there is to know about phylogenetics. Or you can take some of the more advanced, more algorithmically focused computational biology. That won't be the only thing you would cover, but you would cover at least some of the more advanced theory that we can get into in this class for these phylogenetic problems. So as I mentioned at the beginning of the class, I wanted to kind of wrap up with this because it's such an important cross-cutting topic in computational biology. And although I can only sort of scratch the surface of this here, I hope it gives you some of the flavor of some of the complexity that goes into solving some of the hard problems in computational biology, and some of the things you could work on if you actually go into computational biology as a specialty for yourselves. So we've got a couple minutes left, but I think I'm going to stop here because I want to say a few things about the exams coming up and see if there are any questions about that.